In this lesson, we'll be talking about the catalase, oxidase, SIM, urea, and citrate test. Our first test is the catalase test. The catalase test is typically used to see if bacteria have an enzyme called catalase or not. With the catalase enzyme, this is typically an enzyme that aerobic bacteria or bacteria that utilize oxygen have, which allows them to detoxify hydrogen peroxide. So typically the catalase enzyme is going to take hydrogen peroxide and neutralize it or break it down into water and oxygen, both of which are then harmless to cells that the cells can then utilize. So in order to do a catalase test, it is actually quite simple. All you have to do is add a sample of bacteria to a clear microscope slide, and then to that sample of bacteria, you add a drop of hydrogen peroxide. And if the hydrogen peroxide bubbles, like what we see here, that is a positive reaction. So what's happening here is the likely aerobic bacterium has released the catalase enzyme, and when it releases the catalase enzyme, that breaks down or neutralizes the hydrogen peroxide, which causes oxygen to be released, and that induces the bubbling that we see here. And then in the sample at the bottom, when hydrogen peroxide was added, there was not any bubbling. So no bubbling is indicative of a negative reaction. So in this on the slide here, these bacteria didn't have the catalase enzyme. Potentially they're anaerobic, and since they didn't have the catalase enzyme, they weren't able to break down the hydrogen peroxide, which caused oxygen to not be released, and we did not see that bubbling that we would typically expect. And then we just have another slide here kind of discussing the results too, but again, if it bubbles, and the bubbling will occur instantly, but if it bubbles, that's positive for the catalase enzyme. Bacteria able to neutralize hydrogen peroxide and are likely aerobic. If the catalase enzyme is not present, we will not see bubbling, indicating that the bacteria are likely anaerobic, meaning they're not typically don't thrive in oxygenated environments, and they lack the catalase enzyme. The next test we'll talk about is called the oxidase test. So the oxidase test is used to see if a bacterium has an enzyme called cytochrome C oxidase. This enzyme is common for aerobic bacteria and facultative anaerobes. This enzyme allows them to carry electrons through the electron transport chain during oxidative phosphorylation, and as they generate ATP or energy. So typically bacteria that have this cytochrome C oxidase enzyme are considered oxidase positive, whereas bacteria that lack it are considered oxidase negative. It doesn't necessarily, if the test comes back negative, it's not a for sure indication that the bacteria are anaerobic. It just means that in order to utilize oxygen, they may have some other enzyme. But certainly, if a bacteria has the cytochrome C oxidase enzyme, it is able to survive in the presence of oxygen, whether it be aerobic or facultative anaerobe. So to do an oxidase test, you often use something called an oxidase dry slide, which has the oxidase reagent already built into it. So it's actually quite simple. All we have to do is collect a sample of bacteria with a sterile cotton swab, or even sometimes a wooden toothpick. And once we've collected that sample of bacteria, we just rub it into the oxidase, one window of the oxidase dry slide. And then you wait 20 seconds to see if a reaction occurs. If during the 20 seconds the window of the dry slide turns purple, that is a positive reaction, and we would say that the bacteria have the cytochrome C oxidase enzyme. If during that 20 seconds the window does not turn purple, then that's a negative reaction and the bacteria lack the cytochrome C oxidase enzyme. If the reaction happens after 20 seconds, we would not record it, as that can be considered a false positive reaction. So we do want to make sure that we record that reaction within that 20-second window. Our next test is the SIM test, which is one test that looks for 3 different things among bacteria. So SIM is an acronym. The S is for sulfur reduction, the I is for indole production, and the M is for motility or movement. So we're going to go over each of these. So starting with sulfur reduction, some bacteria are able to reduce sulfur, and by reducing sulfur, they produce a gas called hydrogen sulfide. Hydrogen sulfide can be produced by bacteria in 2 primary ways. Either the bacteria are able to convert an amino acid called cysteine into pyruvate using an enzyme called cysteine disulfurase, or in some anaerobic bacteria, thiosulfate, which is the last electron acceptor in anaerobic respiration, when it is reduced, it can result in hydrogen sulfide gas. But in either sense, bacteria that are able to reduce sulfur can produce hydrogen sulfide gas. Why is this important? Well, bacteria that are able to produce hydrogen sulfide gas have been linked to gut inflammation. So a lot of GI pathogens, when they get into the gastrointestinal system and they release this hydrogen sulfide gas, it causes severe inflammation within the gastrointestinal system, which can cause vomiting, diarrhea, cramping, the sorts of things that we would expect amongst GI pathogens, things like salmonella. Now, how are we able to see if sulfur has been reduced and then ultimately led to the production of hydrogen sulfide? Well, hydrogen sulfide is an invisible gas, so you're not going to be able to see it. So what has been done is iron salts have been added into the media, and when hydrogen sulfide, if it's present, binds to the iron salts, it causes a reaction that results in this black discoloration. So how we would know that sulfur is reduced and then ultimately hydrogen sulfide is produced is we would see a black discoloration. So if your test tube turns black, typically 24 hours after inoculation, that's a sign that there is a presence of hydrogen sulfide. However, if it's still all clear, that is an indication that hydrogen sulfide is not present and that sulfur was not reduced. The second part of the SIM test is the indole production component. So some bacteria have an enzyme called tryptophanase, and if they have the tryptophanase enzyme, it means that they're able to break down tryptophan, which is an amino acid, into indole and pyruvate. So how we test to see if this has occurred or not is we add a reagent called Kovacs. Kovacs is typically added about 24 hours after inoculation and incubation. You add a few drops of the Kovacs reagent into the top of the test tube. And if the Kovacs reagent turns pink, that is a positive reaction. And this will typically occur right away. So over here we see a positive reaction as it turned pink. And then over here it's still kind of this clear orangish color, so that's a negative reaction. So again, if it turns positive, that tells us our bacteria must have the tryptophanase enzyme, and with that enzyme they are able to break down tryptophan into indole. However, if your bacteria are unable to cause the reaction to occur and the Kovacs reagent stays clear, that is an indication that your bacteria lack the tryptophanase enzyme and are unable to break down tryptophan into indole. And the last part of the SIM test is the mobility portion. So we can test to see if bacteria are able to move or not, which is an indication of whether or not they have flagella, which are the motor appendage used by bacteria. So in order to do this, we have to kind of first talk about how these plates or how these tubes are inoculated. So we're going to move ahead for just a second. In order to inoculate a SIM test, you have to do a unique procedure called a stab inoculation. So you take an inoculating needle, which is similar to the loop, it's just that at the end there's not a loop, it's just a straight point. But you take an inoculating needle, you would obviously sterilize it in a Bunsen burner or an incinerator. You would let it cool down for 30 seconds or so. You would collect a sample of bacteria, and then you would take that needle and you would stab it straight down into the SIM media and then straight out. You do not want to stir when you do this. You just literally want to stab it straight in and straight out. The reason why you do the stab inoculation is because you want to create something called a stab line. Because the stab line is important when we look for motility. So what you would do is you would do the stab inoculation, and then you would incubate for 24 to 48 hours, and then after that time period is up, you would look at that stab line. And if the stab line is still very thin, such as what we see here, that's a negative reaction as far as motility is concerned. So what that basically means is we stab the bacteria in here and then they just stayed put. They weren't able to move to the side, they just grew right where you put them, which means they're non-mobile and do not have flagella. Now over here where we have this thick stab line, that's a positive motility reaction. So when those bacteria were stab inoculated into the media, they were able to start swimming outwards, swim sideways, and create this thick stab line. So that is an indication that yes, the bacteria are mobile, yes, they would have flagella. So typically on day one, we do these stab inoculations, and then we incubate, like I said, for 24 to 48 hours at 37 degrees Celsius. And then day two, you finish the procedure, which is just looking to see if the test tubes turn black or not, and if they did, then we know that sulfur was reduced, and that was led to the production of hydrogen sulfide. And then we add the Kovacs reagent to the top of the media. If it turns pink, then we know that indole was produced, and that those bacteria have that tryptophanase enzyme. And then we look at that stab line, and we try to decipher it's thick or thin to see if the bacteria have flagella and are able to move or not. Now, in the ones where sulfur was reduced and led to the production of hydrogen sulfide, if the bacteria do have an abundance of flagella and are very mobile, a lot of times that entire test tube will turn black because we kind of have to account for that really, really thick stab line. But in bacteria that are able to produce hydrogen sulfide and are mobile, the stab line will be present. It'll be much thin, so that black line will be much thin as well. And then obviously this over here was negative for motility, and it didn't produce hydrogen sulfide, so we don't see any of that blackness there.